Hello everyone, and welcome to today's webinar, Intact and Peptide Level Characterization of Nanobodies by High-Resolution HPLC-MS. I am Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SCIEX. To learn more about our sponsor, please visit their website at sciex.com. So let's get started. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Peng Yu, Head of Technology at EpiQMAx. Dr. Yu, you may now begin your presentation. Welcome, sir.
Thanks for the introduction. Welcome everybody to today's webinar. I'm going to talk about the characterization of powerful nanobody-based research tools. My name is Peng Yu. I'm the head of technology at EpiQMAx. So before I start, I'm going to briefly talk about who we are. We are a biotech CRO. We spun off from University of Munich over 2 years ago. And we actually have two branches in our company. One deals with histone code profiling to decode epigenetics, and the other is biologics characterization, which I lead. And this is what I'm going to talk about today. And our missions in this branch are to improve the quality of therapeutics via advanced bioanalytics. And we do this via advanced instrumentation, workflows, and software. And together, we offer decades of combined experiences to our customers and solve their problems. And primarily, we work in the following area: recombinant proteins. So this would include the antibodies, and also protein conjugates, such as antibody-drug conjugates, and also, of course, some of the nanobody derivatives we're going to talk about today. And we do most of the work on SCIEX XLC and X500B benchtop QTOF system. When we are under extremely sample-limited case, we can also utilize a triple top system that's fitted with a Nano-LC in the front end. And for today's webinar, we are going to talk about a few things. First, we're going to learn what are nanobodies and their characteristics. And also, we're going to demonstrate this by the production and quality characterization of two nanobodies. Also, in the third part, we're going to characterize the nanobody derivatives. These are the Nano-Secondaries and Nano-CaptureLigand we're going to go into in a minute. So I'm actually going to start by showing this picture. I'm sure most of you recognize right away. This is an IgG-based antibody. It's a multi-domain protein. This is well established. However, it's kind of bulky and complex. So then the question is, are there better alternatives? The answer is an emphatic yes. There are quite a few simpler alternatives, notably the antibody mimetics. There's probably over a dozen of them. Just to name a few here: Antibody, DARPin, Aslin, and Educating. They are in various stages of a therapeutic development from various companies. Then there's also a class of what is called single-domain antibodies, most notably the variable heavy chain antibody, VHH, also known as nanobody. These are from camelids, like camels and alpaca, llama. There is also this variable neoantigen receptor from sharks. And I think nanobodies actually arguably made the furthest progress into clinical development. And actually, in 2019, FDA approved the first nanobody-based therapeutics cavalizumab for the treatment of thrombosis. So, what are nanobodies and their advantages? Right. So I just told you this is actually from the camelid's alpaca here as an example. Researchers discovered a couple of decades ago that alongside IgG-based antibodies, there's also a heavy chain only antibody found in alpaca, and then they went ahead and took the binding domain of this heavy chain only antibody, and it created what is now called a nanobody. It's about 15 kilodalton in size, and that's 1/10 of the size of IgG-based antibody. Because of the really small size, there are actually a lot of advantages. For example, they are highly stable, right? So the melting temperature in the range of 80 to 90 degrees are not unheard of. There is no glycosylation. This means they can be produced in prokaryotic cells, but in the same time, it also means it does not engage the complement system in human beings. And also, it's got a fairly short plasma half-life due to the small size and very fast renal clearance. Well, it can extend this if needed, right, by conjugating this to biopolymers, such as PEGs or some other carrier protein. And nanobody itself is a pretty good ADC carrier. You can conjugate various molecules onto this small protein, as we will see in our next slide. Or actually, the main story of today is about the derivatives of nanobodies, and we're going to be talking about two particular kinds. The first one is nano-secondaries. What are nano-secondaries, right? So nano-secondaries, we go through the same process to create a nanobody, and then now we can actually put a fluorophore, so way of detection, onto this nanobody. And if the nanobody itself is targeted against the primary antibody, then of course this is a secondary nanobody. And because it's a nanobody, right? So in the meantime, now we call this a nano-secondary, as can be seen on the right here. This is a alpaca anti-rabbit IgG nano-secondary. It's got a fluorophore on it, and it's fairly small, about 2 nanometers in size. So here is an illustration of what nano-secondaries can do. It forms a pretty small and precise complex with your primary antibody. But because nanobodies are actually produced recombinantly in prokaryotic cells, so by definition, they are always monoclonal. Whereas polyclonal-based secondary antibodies, they form large and arbitrary complex with your primary target. And with the small size and the precise complex of nanosecondaries, we can do a few really neat things. For example, we can actually perform high-resolution multiplex imaging here, showing triplex. The primary antibodies are from 3 different classes of mouse IgG directed against 3 different target proteins in the same cell. And now we have created 3 different nanosecondaries. So these are shown in different colors, green, red, and white. I think you're actually seeing white is totally in white. Apologize for that. You don't see any writing, but this is the white color. Right, so 3 different flavors of nanobodies with 3 different specificities toward 3 different primary antibodies, who in turn target 3 different cellular proteins. And this allows us to actually image the 3 different proteins sequentially and create an overlaid image, such as the one shown on the left. Right? And again, because their size is pretty small, we can achieve a fairly good resolution with this. So this is for nanosecondaries. Additionally, we are also going to talk about Nano-CaptureLigands. Here we go. So nano-CaptureLigands, these are capturing reagents used to immobilize target antibodies in biosensors or in ELISA assays. And they actually carry bulky moieties. This can be captured by streptavidin plate or by a streptavidin coated biosensor, such as shown on the right here. All right, so what we're looking at here is a biosensor. The biosensor body itself is depicted in the white cylinder. It first captures the nano-CaptureLigand by way of streptavidin bulk interaction, and then the nano-CaptureLigand is now free to capture your primary antibody of interest. And once that is done, now we're ready to study the interaction between your antibody of interest and its ligand. So shown on the next slide, it's just what we did, right? So this is a biolayer interferometry. You can actually perform similar experiments using SPR or surface plasmon resonance. What we're looking at here on the left side, right? So first we load the nanobodies or the nano-CaptureLigands onto the biosensor. You can see the loading curve and establish a baseline. So we know that the loading is complete. And now we're going to load the primary antibody onto the biosensor. Again, we see a loading curve, and then we'll achieve our baselines. So right here. So now we are ready to perform our real experiment. You can wash on your ligand, and at different concentrations, this is the different curves on the association stage, right? So they are at different heights because they have different concentrations of the ligand. And then once you're done, then you wash it off, get K off, and then you can calculate the KD from K on, K off. Something I'd like to point out is, let me see, right, so BI, what this I just said, right, so biosensors do require a capturing reagent to immobilize your antibody of interest or your protein of interest. And what I like to point out is that look at how stable the capturing are, right, at different stages and also during the association stage. This is pretty useful. So we spend all this time talking about nanobodies and their different useful formats as research tools and their derivatives. Now we're ready to move on to the production and the characterization of them. And we're going to take a look at two examples. They are both produced in nano-secondary format and also a nano-CaptureLigand format, with a nano-secondary format containing two extra engineered cysteine residues. Here is a summary. Right. So these two nanobodies, VHH1 and VHH2, they have two different specificities. And then these are the naked protein, right, expressed recombinantly in prokaryotic cells. These are the, again, the C at the end stands for the capture ligand format. And we also have the VHH1S and VHH2S, the nano-secondary format. And these guys have the extra cysteine, or actually it's the three style groups that we are after. Right, so these are naked nanobodies, naked proteins. They are not ready to be used as capturing reagents or secondaries yet. So in order to create the nano-CaptureLigand, we are going to react the nanobody with NHS biotin reagent and then derivatize the nanobodies to put a biotin label onto the proteins. And in order to create the nano-secondaries, we are going to react this with lux fluor malabine reagents, and this is targeted to the style groups on the extra cysteines we put in. All right, so that's all clear. And then we are first going to take a look at the qualities of the proteins itself before we move on to the capture and secondary reagents. Right. So the ID and the purity confirmation, we did a quick LC-MS acquisition of intact mass of VHH1C as an illustration. On the upper panel, it seems there is a peak splitting, which I'll explain in a second. Even from this, we can actually conclude it's fairly pure. It's more than 99% pure. And on the lower side, we're seeing the masses of this particular nanobody. And the reason why we see so many different patterns or so many different clusters is because they simply represent different charge states, right? So we can actually zoom in on one of the charge states. Let's do so. Voila. Right, so I think we zoomed in on the 10 plus charge state. So there are indeed two clusters. This actually explains why there are two peaks in the LC illusion profile on the left. And we can do a reconstruction to get their impact mass. And we did get one peak at 13,720 roughly. So this matches pretty well with the theoretical mass, right? Very close. However, there is a second peak that's about minus 17 Dalton away from the main peak, if you will. And this actually simply is the glutamine to pyroglutamate conversion, a loss of 17 Dalton. This happens spontaneously in vivo and does not affect protein function or anything. So there's nothing really to worry about. It just happens, natural process. And we can, of course, also zoom in on a particular isotopic distribution. This is actually reconstructed, right? So what we see here is an overlay of experimental distribution measured in blue and the theoretical distribution, calculated distribution. It's actually a fairly good match, I would say. And we get near baseline isotopic resolution. But we can, of course, always, not always, but in this case, it's actually easily achievable baseline resolution. Just by tweaking a couple of instrument parameters. And we did this, of course, for all 4 of the nanobody proteins, and were able to confirm their ID by the difference between observed mass and theoretical mass. So I say they all look pretty good. And of course, we are interested in ID, but also in other quality attributes. This would be additional product quality attributes, primary sequence and disulfide bond, and also post-translation modifications. Additionally, in terms of or in case of a label proteins, we're also interested in the localization and the degrees of labeling. This is known as drug-to-antibody ratios if you are from the antibody-drug conjugate world, if you are coming from ADC. These attributes are assessed at peptide level. Right. So what we do is take the protein and do a digestion, usually by trypsin. Of course, you can use some other protein. The illustration here is shown for an IgG. This is what we will do for IgG. But in our case, for nanobody, the process is exactly the same. And since we're interested in disulfide bonds, we just have to do this digestion under non-resistant conditions to preserve the disulfide bond. Okay, so let's take a look at one example. This is a VHH1C. So here again, we are still looking at the protein. We did the digest. We run the protein mixture through LC-MS, actually LC-MS. So if you are from the field of proteomics, This may look awfully empty to you, right? So you may even wonder if this is a blank. Actually, remember, we are only looking at a protein digest, and the protein is only 15 kilodalton. So there's only 10 to 20 peptides in there. They should look like this. This is exactly what we are expecting. And then using Biopharma View Flex software, we can identify all the peptides and then map the peptides back to the sequence. What we are looking at here is the green bars on the bottom, and the sequence itself I actually blanked out because the sequence is proprietary. The green bars, these are peptides identified via MS, and again, they are mapped back to the protein sequence. This is fairly analogous to next-gen sequencing, right? So you get your short DNA or RNA reads and then map back to your reference genome, and we are doing precisely the same thing. Additionally, did I just talk about the disulfide bonds, right? So this is shown in magenta color. Can you see? Here we go. This is the disulfide bond still intact, and we can see the two blue bars highlighted in blue, and this magenta thing, this bridge of these two peptides. This indicates this is where the disulfide bond is. Good to see. And because of the prokaryotic expression and the presence of a disulfide bond, right? So then this actually requires some special expression conditions because of the redox potentials in the prokaryotic cytoplasm. And X500B being a QTOF, we can also get MS information, and this is what I mean. Right? For each and every peptide, not only do we identify by MS1, we also identify by MS2. Okay, so we're looking at a fragmentation pattern. And the way to interpret this is that if you are not from the mass spec business, or if you are not from this, if you haven't spent the past 10 years using mass spec or looking at the mass spec data, essentially what I'm trying to say is I'm going to briefly walk you through how to interpret this or what we are looking at. So first of all, we find our molecular ion. Yeah? This is M-18. This is simply a loss of water, right? So this represents the peptide, the molecular ion. And then we can see a series of other ions. For example, Y6. This simply is a peptide with one N-terminal amino acid missing. And then there's a Y5. This is a peptide with two amino acids missing. And then we go to Y4, Y3, all the way to the end. We can, of course, also read this the other way, right? So we can start from B6 here. This is the peptide with one here, with one C-terminal amino acid residue missing. And then there's B5. So this is the peptide with the two amino acids missing. And of course, going all the way to the other end. So this, by doing this, right, so we not only get peptide resolution, we also get amino acid residue resolution. This would be especially important in sequence variant analysis, where we would need this amino acid residue resolution. Let me then take a small detour to HCPs, host cell proteins. Host cell proteins are process-related impurities that resulted from prokaryotic or eukaryotic expression, and this should be removed, but very often the removal is incomplete, and then this resulting in the host cell proteins. They can affect the efficacy or safety of the therapeutic, so they should actually be very tightly monitored. And then we actually decided to take a look at this because the sample is there, the process is already done. So what I mean by that is we did a trypsin digestion of our sample, and that would actually digest all their proteins, regardless if it's the protein of interest, meaning regardless if it's the protein that you expressed or if it is a host cell protein, right? So the process is done, then we can simply ask the question, are there host cell proteins to be found in that particular sample? And we actually, let's see, yes. So we actually pooled two samples just to increase the coverage, or actually to maximize the chance that we see some host cell proteins. And yes, we do see them. They are about 100 ppm in abundance, based on a rough estimation from peak intensities. And then let's actually come back from the detour and just talk about the production and the characterization of the nanobodies themselves. Right? So now we're ready to look at the nanobody derivatives, namely the two reagents we're going to cover. So again, right. So again, this is a summary of what we have been talking about and what we're going to talk about. Just now we talked about the four next proteins, remind you, and then next up we're going to talk about the real reagents, the Nano-CaptureLigand and Nano-Secondaries. So first, we take a look at the VHH1C biotin. This is the Nano-CaptureLigand, and also the Nano-Secondary on the right. We're looking at the elution profile. On the left, there seems to be 3 peaks, right? You can probably make out at least 3 components. And on the right, it's fairly homogeneous, I would say. There's only one major component. And then when we look at the mass, on the left side, we can actually clearly make out 4 clusters. On the far left, 13,702. This is the nanobody by itself with no labels. And then next one up, 14,193. This is the nanobody plus 1 biotin label. Next one up, this is plus 2 biotins. And yet next one, this is simply with 3 labels. Whereas on the right side, the Nano-Secondary, we can actually see only one major peak, 16,000 or so. Right? There is a small cluster that represents the nanobody plus 1 Alexa fluor. By the way, this is 16,000, and this is a nanobody plus 2 Alexa fluor labels. And of course, there is a very tiny, actually you don't see here, but it is present, super tiny peak. That is a naked nanobody without any labels, super tiny. And using this information, we can calculate the DAR distribution. Let's see. Here we go. Looking at the NHS biotin conjugation, we can see a distribution, right? So they are not as homogeneous as the one on the right, whereas where most nanobodies carry 2 labels, where on the left the Nano-CaptureLigand, some of them carry 1 label, some carry 2, and the mean DAR is a little less than 1. And on the right, so we achieved almost 100% labeling with a mean DAR of 1.9. The maximum we can get is 2. And we are not only interested in the DAR distribution and mean DAR, we are actually also interested in where the labels are located, meaning the exact residues. So we can do the same exercise as we just did before, meaning the peptide mapping exercise. And we did this. We did the digestion. We run them through LC-MS, and we use the software to find out where the peptides are. and also identify where the labels are. Okay, so what are we looking at? Each red dot on any green bar, or each red dot on any peptide sequence represents a label. Here, in this particular case, would be either biotin or Alexa Floor. This is for the, excuse me, this is for Nano-CaptureLigand in some water. So we see that there are 6 labels, whereas for the Nano-Secondary, VHH1S, we see the labels are precisely where we put them on both ends. Excuse me. Because HcAbs is a quantitative, we can also calculate the degree of labeling on each site. There we go. So this, if you come from the world of proteomics, this will be known to as a site occupancy. First, we look at VHH1C. This coupling is done by N-hydroxyl-6-cinnamide. They target 3 amino groups on the side chain of the lysine, and then we can see that mostly it's on 6 lysines. Actually, I have to say, before this analysis, it was expected that most labeling would fall onto K5, K6, and K7. There is one additional lysine in there, at least one. But however, with the data, we can actually show that there is a significant labeling on K3, right, and some labeling on K1. So that tells us not all lysines are created equal. And whereas for VHH1S, the labeling here is done via maleimide, and it's quite specific toward the diol group. And I think another reason for this specificity is simply because there are only 2 free diol groups that we put in through protein engineering. And it's very, very nice to see that the labeling is pretty close to completion. And since the NHS chemistry has some selectivity, or in other words, the reaction didn't really go to completion, or yet in other words, there may be some room for tweaking. And then we thought we can actually tweak it, and we did this. Here's the result. So what we did is to vary the NHS biotin reagent to nanobody ratio, and simply change the labeling pattern. Now I have to say that it's not the more label, the better it is, because if you put too much label on, right, then this can have a negative impact on protein structure or binding properties. So we don't want to label this excessively either, but of course we do want some labels to be on it, right? So it can be captured, so itself can be captured by structivity. This is what we see. By increasing the reagent to protein ratio, we can drive the mean DAR from 0.9 to 1.8 to 2.3, and then we can also shift the distribution. This is actually, how do I say that, pretty nice to see. And using this LC-MS data, right, so we can really precisely define how our product is going to look like. In summary, what I just talked about is Nanobody is a pretty robust alternative to IgG-based antibodies, and because of their size, there's a whole bunch of properties that came with the small size. And also, we can produce recombinant nanobodies with a defined specificity. And again, because they are recombinantly produced, they are always monoclonal. And LC-MS allows a very facile and precise characterization of the quality attributes as I've just shown you. And lastly, we can produce a very versatile nanobody derivatives, such as the ones in this webinar, Nano-Secondaries and Nano-CaptureLigand. There are additional formats you are welcome to check out. Check them out. And moving forward, we are planning to assemble a comprehensive and standardized assay portal for nanobody characterization, and this would of course be also applicable to to other simple and small antibody mimetics or other single domain antibodies. And then using the MS data, we can further optimize the nanobody products. So now I'm going to come to perhaps the most important slide. My co-authors, Julia and Kerstin from SCIEX, they spent a lot of time with me to really get me up to speed with the instrumentation and software. Todd is a true pro in preparing webinars. If you like this webinar, this is because of Todd. If there's something you didn't like, this is because on that particular place, I didn't agree with Todd. And also, Christian and Felix from Comatech, they provided all the samples and also all the slides for Nanobody, the introductory slides for Nanobody. And Frederika and Victor from my company, from EpiQMAx, our team, they did a lot of sample preparation and data analysis respectively. And last but not least, I would like to thank you for your attention and staying with me so far. You can reach me via a variety of different ways. I'd love to hear from you. And now I'm going to open the floor to questions.
Thank you, Dr. Peng, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. So Peng, our first question, what do you mean by bulky and complex related to its applications compared to the nanobodies?
Oh, thanks for that question. It's actually a really great question because here we are really talking about related to its applications, right? So then I would consider in certain applications, for example, the ones we talked about, just for capturing or as a secondary antibody, I would say IgG is quite bulky, right? It's really not super necessary to have a large molecule. Whereas in other applications, for example, if you are playing with the complement system where you do need the glycosylation, or if you are creating some bispecific or I don't know trispecific, then it's actually quite useful to have the IgG. But again, it depends on the application. And yet in some other scenarios where you just need a target blocked, then I guess a nanobody would actually do just the same job and still being a smaller molecule. So I hope that somewhat clarifies this question.
Wonderful, thank you so much. So moving on to our next question, what are some advantages of nanobodies over antibody mimetics?
Nanobodies over antibody mimetics. Okay, so I guess I got to be careful to not mess this one up. Nanobodies, I would think nanobodies because nanobody itself is a derivative of a naturally occurring antibody, right? So then this means it's already got some pretty good natural affinity toward different targets. And then you can, of course, do some antibody maturation in vivo in a camel or I don't know in a llama, etc. Right? So whereas some of the antibody mimetics, you have to do this part in vivo. I guess there are probably some more things that I'm not aware of, or maybe somebody else have a better idea. And yeah, so I'd say if I come up with something new, I can email you.
Great, thank you so much. We do want to thank our audience for all these great questions that are coming in. So how about our next one? Let's take a look. Okay, here's a great one. So how much optimization for the LC and MS method did you have to perform? And the second part to the question is, were the acquisition methods similar to monoclonal antibodies?
For the first part, I would say really not that much, because at the end of the day, a nanobody is still a protein, right? So we can just take the or actually, what I did, take a generic method for those for proteins of that size, and then just run. It usually is pretty good. Very rarely do you have some how do I say that, some weird phenomena where you have to troubleshoot. But most of the case, a generic method works really well. And as far as monoclonal antibody goes, I think there, because of the size, right, so this is the primary matter, then we would have to use a much higher mass range. And also, because of the size, we would have to change the sensitivity versus the resolution trade-off toward a higher sensitivity, because a huge molecule, the signal is really dilute out because of different isotopic combinations. So yeah, so for nanobodies, we can go with higher resolution. Mm-hmm.
Great, thank you so much. Excuse me. So there was one point that you talked about getting a higher isotopic resolution. So how did you do this?
Yeah, so essentially, I kind of alluded to this in the question prior to this, right? So I think actually in most modern instruments there's a possibility to get a higher resolution at the cost of sensitivity, right? So if sensitivity is more important to you, then you drop resolution. If resolution is more important to you, you would actually drop a little bit in sensitivity. So I think particularly on SCIEX instruments, this parameter is called time beams to sum. So you would actually increase this number to get more sensitivity, but if you want more resolution, you would decrease time beams to sum. And then of course on other types of instruments, the particular parameter to tweak is slightly different, but generally this is how you would go about it.
And it looks like we have time for a couple more questions. So now, Peng, are you expecting a change in ionization or the intact molecule naked versus different labeling amounts?
I would say this is a good question. I would say if it really depends on how big of a molecule that is, and if the molecule itself is heavily charged, etc. Right? So if the molecule is relatively small compared to the protein, then I don't expect this to be a major impact. However, if this molecule is relatively big compared to the protein, then of course you can't be sure, because then there could be a little more variation. So the answer really, hate to say, depends.
Not a problem. So we do have one audience member asking, I was wondering about your HCP detection sensitivity. What would be LLOQ of HCP for your setup to see?
What would be the LLOQs? Honestly, I cannot give you an exact number, right? So the number I provided in the slide, this is just an estimate. And as I said in the slide, I just want to take a detour, right? So I'm going to guess we probably can go down an order maybe, but honestly, I I do not have a good answer to this question. But if you are interested, we can go offline and have a more in-depth discussion.
Great, thank you so much. Now it looks like we've got time for one more question. So Peng, here's our last one. Can you reduce the existing disulfide bond in the nanobody protein and bond electrophore to the resulting free thio groups?
Well, one would think this would be a great idea, right? So you don't have to put in the extra cysteine residues. However, if one were to do this, then I think it would also very likely negatively affect the binding properties of antibody and also the properties of the nanobody itself. It may not be as stable. It may, I don't know, it may not bind the correct target, etc. So yes, you can, but I wouldn't.
Great, thank you so much. So we want to thank you for your presentation, Peng. Do you have any final comments for our audience before we go?
Actually, I would say thanks everybody for attending. I do appreciate staying with me so far. And again, you're welcome to check out our website or get in touch. My contact information are here. And yeah, and lastly, I would like to say thanks SCIEX and Navruz for hosting the webinar. It's been a great pleasure.
Thank you again, Peng, for your time today and your important research. We'd also like to thank LabRoots and our sponsor, SCIEX, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you again, and bye-bye, everyone.